Bonjour, je suis ravie d'être ici parmi des collègues et je parlerai en anglais parce que c'est un peu difficile pour moi de parler le français scientist, right? Okay, so I'm changing gears and I'm talking about toxicity of TTR in FAP, familial amyloidotic polyneuropathy. I won't say at this time too much about the disease because people here know about it, and you will spend the whole day talking about clinically aspects. Just to remind you, this is an autosomal hereditary and neurodegenerative disease, a late onset. There are more than 100 mutations known. It is a systemic amyloidosis with special involvement in peripheral nerves, giving rise to a sensitive and autonomic neuropathy. The most common mutation, it's a substitution of methionine for valine. How do I advance this? Okay. So you see there are amyloid deposits in peripheral nerves that you see here very well. And so through structural studies, we have here, it's a tetramer of 4 identical subunits, which you see here, a monomer plus a monomer making a dimer, and the dimer assembles with another dimer, forming a hydrophobic channel, which is the place for binding thyroid hormones and other drugs and ligands, as you will see later. We know now through many different types of studies in many labs that this tetramer circulates in blood, especially with mutations. It gets unstable, dissociates into monomers, and these monomers then assemble into ribbon-like structures, making like a tubule. Which is a fibrillar material. We have in the lab in the recent years specializing more, and our focus of research has been this structure and how to get rid of this structure. Okay. And so we have several, these structures, fibrillar or prefibrillar, can act on cells in different ways. You can have internalization way in which the structure can have receptors or not, we don't know yet, can enter the cell, go through the endosome, and then be degraded in lysosomes. So you can get rid of these structures by lysosomal entrance and degradation. The other way they can be toxic, it is through transduction. So you have internalization versus transduction, and in transduction we have studied this receptor, which is the receptor for advanced glycation products called RAGE, and this receptor recognizes amyloid structures. It recognizes AA, Alzheimer, etc., the A-beta peptide, and it's a transmembrane receptor. The cytosolic tail is going to trigger a kinase cascade and is going to activate NF-kappa B. And NF-kappa B then can activate products like inflammatory cytokines and oxidative stress molecules. And we have come to several mechanisms. I mean, they are models, okay? We think they work. And this has been, we have taken back and forth cell culture studies. I won't show two cell culture studies, but, and we go to animal models, and then to FAP ex vivo FAP tissues. So it is this triangle that led us to the conclusions I'll show you next. So ex vivo studies, we had access to biopsies, FAP biopsies, from asymptomatic patients collected now 30 years ago, many years ago, at Hôpital Saint-Louis in Porto. And so we had different biopsies. We had normal, and when I say normal, are from siblings of asymptomatic carriers. At the time when these biopsies were collected, when there was no diagnosis, a molecular diagnosis, so the pathologist did not know, only saw amyloid, but he did not know if it was TTR or not, because it was not described. So that's why we have these biopsies. So at the time, they were Congo red negative, the normal, and what we call FAP0. So there were no clinical symptoms, and then, of course, they were clinical symptoms. So what we did at the time, around 2000, we stained these biopsies for TTR. And staining for TTR, we found in FAP without symptoms, okay, FAP0, we found immunoreactivity with the TTR antibodies, which you see here, though they were Congo red negative. And of course, you have reactivity in case you have Congo red. And the electron microscopy with the gold particles, TTR gold, anti-TTR gold particles, you can find that this picks up amorphous material that is not yet totally fibrillar. So they are protofibrils that don't have the characteristics to bind Congo red, but they are already there. So they are oligomeric TTR structures. Okay, so we concluded that TTR is aggregated non-fibrillar material very early in FAP. Some of these cases, by the time we studied 20 years later, were still asymptomatic. Okay? So these aggregates and oligomers can be in the body, nerves, everywhere, many years before clinical symptoms. So they are called, so they are aggregates. This I had already explained, knowing that we studied not only fibers, but as well aggregates. These aggregates, they also bind RAGE and elicit this kinase cascade, giving then transcription, activation of transcription. And this was shown in FAP tissues. I didn't bring images, but you see activation of NF-κB, and also in cell culture. And you can, I'm only going to show this one. So this one, whoops. So in this one, you can see in FAP0, activation of synthesis of interleukin-1 beta in the nerves of FAP very well, you see, and also by in situ hybridization. So that means the nerve is synthesizing EL1. Concerning animal models, we have developed over the years transgenic mice for methionine 30, and we cross these mice with heat shock factor 1 knockouts. And this way we achieved not only deposition, prefibrillar deposition in nerve, but also we see if we stand for IL-1 beta, we see both nerves. IL-1 beta is activated not only in nerve, but also in dorsal root ganglia, DRG. These mice, so this is the problem now. Suivant, I activated not something. Okay, I found how to do it. Okay. So having this access to this animal model, we were able to have material like dorsal root ganglion that is not accessible otherwise. And what you see here is an investigation of ER stress. Oh, again, I don't know, but anyway. So you have here stainings and searching for other toxic mechanisms. And one ways of cell death, it is through ER stress. And so we stain for the master switch of ER stress, which is this molecule called BIP. And you have here a dorsal root ganglia without TTR deposition. And here, dorsal root ganglia, you see here the sensitive neuron with TTR around it, extra cell here. So if you have TTR deposition, BIP is activated, the concentration increases. And if you stain by confocal microscopy, you have in green the TTR oligomers, and in red, you'll see that BIP is activated inside of neuron. And we have seen this activation of BIP not only in animals, but also in humans. We studied at that time salivary glands. And in salivary glands, BIP is, you see very well that BIP is activated. So, to show you a long story in a slide, okay. We have studied many looking for intracellular dysfunction in FAP, and what you see here. We have found evidence for several pathways, and okay. So you can have rage, as I explained to you, and FKB, several, the transcription, including metalloproteases, MMP. The mitochondria in our hands, and so far, is not a major activation of the pathway, death pathways, though we find caspase 9 and caspase 3. We also had some evidence through FAS, activation of FAS, caspase 8, and then apoptosis. We had oxidative stress. We could see different molecules, proteins, lipids, that are activated in these pathways, and we saw calcium deregulation as well. We saw that ER is going to release calcium through rhodin receptors, And we look for the different branches of ER stress, in particularly this activation of PERK, which is a kinase, PERK kinase, that phosphorylates this translation factor, EI2 alpha, which is phosphorylate, and then is going to activate caspase 12 and 3, and then leading to apoptosis. We don't know. Certainly, these oligomers are going to damage membranes. We know that's as well a way of releasing calcium, and so you can have problems here with calcium. And there might be other receptors that we are still looking for. So anyway, we had concentrated and tested different hypotheses, namely ER stress, and as well this transcription of inflammatory cytokines and metalloproteases. So we know there is an extracellular matrix deregulation. Metalloproteases are activated. And so we had developed combination therapy to see if counteracting ER stress, which we did by a biliary salt called TUDCA, tauroursodesoxycholic acid. It's not the ursofalk, but it is conjugated. So TUDCA, and without telling you a big story, we know that doxycycline, an antibiotic, is going to destroy amyloid fibrils. So we decided to try in mice this combination therapy, and this is what we found. These are transgenic mice. The GI tract that is affected in these mice. We have control, treatment with control, doxycycline and TUDCA alone, but when you put both, you do a combination, you can get rid of deposition. You won't have more deposition. If you lower the dose to a clinical dose, then you also get rid of these deposits and less TTR, less BIP, less ER stress markers. I won't be talking more about the clinical trials going on, because I think later in the afternoon you might have some of this. So the theme is clearance. So we have doxycycline that is now an orphan drug to treat amyloidosis because it destroys amyloid fibrils. Also in AL amyloidosis, beta-2M amyloidosis, and so it's a way of clearance of amyloid fibrils. But cells, the body, as I said, can also take care of this clearance. And so recently we studied in detail. We saw clearance. We saw both fibroblasts and macrophages full of TTR, okay? And this was in skin of transgenic mice, skin of FAP patients, domino transplant patients, and so on. So TTR is eliminated by cells in the body. And here you see a study with fibroblasts and macrophages where you incubate with oligomeric TTR, which is fluorescent, and you merge with markers for fibroblasts or for macrophages, and you see that TTR goes inside. So that is a way of getting cleared. What we didn't understand is that for many years, we don't know why this is now nerve. Why you have deposition in the nerve, fibrillar, lots of fibrillar material, but yet, if you look at these nerves, there is a lack of cellular infiltrates and chemokines. So there is no way to get rid. You only have resident macrophages, of course, but you don't have an answer to get rid of these, of this material. And so maybe we need to understand better why in nerve you don't have cells to get rid of deposited material. And so we went on in transgenic mice and decided to do an inflammatory stimulus. So we did nerve injury, we did a lesion in the nerve of these animals, and this is the inflammatory stimulus. So usually in normal mice, you would have cytokines, chemokines, and so on, being attracted to the site of injury to clear up the debris. We know that, and different molecules, neutrophils, and so on. And what we saw, of course, we saw different molecules being increased, like EL1 or so, and we saw, and I'll show the pictures, that this stimulus promoted incredible deposition of TTR in the nerve. This incredible deposition, we think it affects Schwann cells, so we think these Schwann cells are deficient and cannot activate the inflammatory response and cell infiltrates. Because we don't have, again in this model, we don't have an answer in chemokines as we have in a control mice. So if you do control mice, you have all these molecules, but in these mice, you don't have this inflammatory response going on the same, reacting the same way. So you have a lot of aggregates that are impairing the chemotaxis that we should observe. Okay, and here, so we conclude after some studies that Schwann cells could synthesize TTR. Okay, TTR is mainly synthesized by liver, choroid plexus, pancreas of brain, pancreas, and under this stimuli, so you have your wild-type mice and transgenic mice. So you see some synthesis of TTR in wild-type mice after the crush, which you have here. So you have the sham, the other side, and the injured nerve. But when you go to a transgenic mice, it is really, well, first of all, the mice have more TTR to start with, but in an injured mice, this is huge. You don't see this reaction in liver, okay? It's only in nerve that you are seeing this is happening. If you remove, if you apply siRNA, siRNA has a targets liver TTR, not other organs, but application and treatment can get rid of extracellular deposition. So the main source of TTI is liver. If you shut it down, you won't have as much of the precursor TTR, and so the body removes TTR. But you remove TTR, but look, you still have synthesis in Schwann cells. We know it is in Schwann cells because if you label for TTR with OCT6, which is a marker for Schwann cells, you have colocalization. So you can have, this is the main concern and the main message, you can upregulate TTR, human TTR synthesis after injury by Schwann cells, the Schwann cells that then have unimpaired chemotactic response. And so we have tried recently, I mean, this was published 1 month ago, that what about if we treat transgenic mice, now this is without injury, regular transgenic, our transgenic mice, with an antagonist for IL-1, okay, because so this is one of the inhibitors we treated with, and Akira would prevent IL-1 effects. And after treatment, so we treated young mice. We only know this works on young mice. So you have the control. You see deposition, no treat in sciatic nerve and dorsal root ganglia as well. After treatment with an Akira, we don't have deposition. We don't know, probably we are inhibiting synthesis as well. So blocking this prevents TTR deposition in the peripheral nervous system. What is most important, and we were surprised by that, is that we have regeneration in the nerve. Okay, so we measured unmyelinated and myelinated nerve fibers, and we can see, you see here the control. Look here at unmyelinated fibers that go up. So we tried already doses ready to go to the clinic. So this happens with doses that can be tried. And we are now performing array data to know really what is the mechanism behind these findings, what molecules are activated under these conditions treatment of with Akira. So I hope to have other news in 2 years or so. It takes time. And then the most important persons in this work. So the lab in Porto, the so-called amyloid lab. We have other labs in the group with different people. And of course, this is a work of many years, many people and investigators and support. Thank you very much.